OMG, let's revise 1.3 digestion. So have a look at the following 10 questions. Okay, pause the video and answer it as best you can. Okay, so question number 1 was to define digestion. So that's the breakdown of large insoluble molecules into small soluble molecules that can be easily absorbed. Question number 2, peristalsis takes place in the esophagus. 3, the function of the pancreas is to produce and release digestive enzymes. The function, for question number 4, of bile is to neutralize stomach acid and emulsifies fats. Okay, so it goes from large fat droplets and breaking those down into smaller fat droplets. Number 5, bile is produced in the liver and it's stored, number 6, in the gallbladder. Number 7, 3 digestive enzymes are carbohydrates, so amylase, lipase, and protease. Number 8, describe the function of each of these enzymes. So carbohydrates convert starch into glucose, lipase breaks down fats into fatty acids and glycerol, and proteins breaks down protein into amino acids. For the last 2 questions, so 9 and 10, okay, absorption of nutrients take place in the small intestine, and where is digestion completed? Digestion is completed in the small intestine. Okay, so on to some past paper questions about digestion. Pause the video, have a go at these, and then we'll go through the answers. Okay, so first question in part A, why do we need to digest large food molecules? And that's to make food soluble. In part B, we have a graph showing the rate of digestion of lipids by lipase under different conditions, and we're asked to describe the effect of pH on a rate of digestion of the lipids. And the rate of digestion increases when pH increases. And we can see that as we go up. A higher pH with alkaline solutions is a higher rate of digestion of lipid. Explain the effect of bile on the rate of digestion of these lipids. So 2 marks here. The first mark saying it emulsifies fats from large to small droplets, and the second mark increasing the surface area for lipase to act. Okay, so across to the second part, and we're asked to label where bile is made, and bile is made here in the liver. Okay, and in part 2, bile is stored in the gallbladder. Okay, so you can see how I've labelled those there for 2 marks. Next set of past paper questions, pause the video and have a go. Okay, so in part A, it says, name the process being described that's the muscles in the front of the food relax whilst the muscles behind the food contract, and that's peristalsis. In part B, we have increase in pH versus, on the y-axis, mass of food undigested, and it asks us to state what happens to the mass of undigested food from pH 3 here to pH 6. Okay, so 2 marks, so we'd say it increases until pH 5 at this point, and then for the second mark, it's horizontal, it doesn't change, it's still 16 grams, so it levels off. Part 2, state the optimum pH of this enzyme. If we look at pH 3, here the mass of undigested food is the lowest at 4 grams compared to the rest, and therefore that is the optimum. Okay. In part 3, we have name the organ in the human body where this enzyme is found and name the class of food it acts upon. So the organ is the stomach and the food substance being protein for the second mark. Okay, across to part C, we've got a graph with increasing temperature versus the rate of reaction, and we are asked to describe the effect of temperature on the rate of reaction between 10 Celsius and 50 Celsius. So 3 marks here, 3 points, saying that as temperature increases, the rate increases, okay, to the optimum for the second point, which is at 35 Celsius, and then it falls or decreases back to a rate of 0, okay. The final part here, so 2 mark question, explain why a denatured enzyme can no longer work. The first is say active site changes shape, and the second that the substrate cannot bind. Final set of past paper questions here, pause the video and have a go. Okay, so with the match-up task, you have to get all 3 correct for those 2 marks. So protein is matched to amino acids, starch is matched to breaking down into glucose, and then fats to fatty acids and glycerol. Why is it necessary? It's to be absorbed into the blood. And state the function of carbohydrate foods in the body. They are a source of energy. Okay, so in part B, we're given a diagram showing the digestive system. We're asked to place the letters that identify with the pancreas, which is F, and the large intestine, which is G. Okay, so the two letters which show where protein is digested, so we know it's C, which is the small intestine, and E, the stomach. Both have to be correct for that mark. Okay, so completing the table, the food substance is glucose, the test is Benedict's solution. If the test solution is Burette's, we know that tests for protein. Okay, both of those for 2 marks. And at the bottom, the following table about food tests. So starch, okay, and then iodine, which starts off as orange, but then it goes with a positive test of blue-black. Glucose, okay, is tested for by Benedict's solution, it's the same above. Starts off as blue, and then it goes brick red with a positive result. And that's the end of those past paper questions, end of the video. Well done.